Here we have a schematic of our chromosome, and we are going to be looking at 3 related loci, starting with color, presence or absence of the tail, and fur length in the corgi. So in corgis, red fur is dominant over tricolor fur, no tail is dominant over having a tail, and short fur is dominant over fluffy fur. For our P1 cross, we are going to take a homozygous dominant female and breed her with a homozygous recessive male. And below the pictures, you can see what their chromosomes would look like. Our F1 generation, we're going to get a heterozygous animal at our 3 loci. So you can see to the left what the chromosomes are going to look like. And even though the offspring is heterozygous, it's going to have the phenotype of the mother because that's what it got the dominant alleles from. So it's going to be red, it's not going to have a tail, and it's going to have short fur. So now we have our heterozygous F1 generation, and we're going to take a female from that litter, and we are actually going to do what's called a test cross. And in the test cross, we are going to breed this heterozygous female to a homozygous recessive male for the 3 genes that we're interested in. And the reason why we do this is that it ensures that the alleles given by the non-test parent, in this case our heterozygous female, are actually going to determine the phenotype of our offspring. So it's letting us see what gametes she's going to produce. So if recombination does occur in our heterozygous female's gametes, there's several things we could see. First, if we saw a single crossover between the R and L loci, we would end up with little r, big L, big F, and we can also end up with big R, little L, little F. If a single crossover event occurred between the L and F loci, we could either get big R, big L, little f, or little r, little L, big F. Now, if a double crossover event occurred, we would get little r, big L, little f, and big R, little L, big F. The map distance is equal to the percentage of recombination between each loci. So in our case, the map distance between L and F is 7.5 centimorgans, and the map distance between R and L is 19.3 centimorgans. For our coefficient of coincidence, or C, we are going to assume that it's equal to 0.7. Alright, so there's 8 different phenotypes of offspring possible from our cross. And something interesting you'll see is that the first 2 phenotypes listed, they just look like the parents, and that's not a coincidence. These are the parental types, or the gametes where no crossover occurred. And if you look at the probability, these have the highest probability out of any of these gamete types. And that makes sense because it's less probable that you will have a recombination event. So if we go down to the next 2, we are going to see our double crossover gametes. And you'll notice these have the lowest probability of all because getting 2 crossover events is less likely than getting a single crossover. If we go down again, we're going to see our 2 single crossover region 1s. And we have a slightly higher probability at 9%. If we go down again, we're going to see our single crossover region 2 gametes. And you might notice that they are a bit less likely at 3%. Alright, so let's do a little test and say what's the probability of getting a tricolored puppy with no tail and fluffy fur. So first, we need to figure out the genotype, which is going to be little r, big L, little f. Then we need to figure out the probability of getting that phenotype. And the probability, as it turns out, is 1/2 of the probability of getting a double crossover. And the reason that it's 1/2 is because the other scenario in which a double crossover happens, we aren't going to get little r, big L, little f. We are going to get big R, little L, big F. So to account for that, we need to divide our double crossover number by 2. And if we want to ask what's the probability of a red puppy with a tail and fluffy fur, we do the same thing.